Marburg virus is a hemorrhagic fever virus of the Filoviridae family of viruses and a member of the species Marburg virus. Genus Marburg virus. Marburg virus. MARV. Causes Marburg virus disease in primates. A form of viral hemorrhagic fever. The virus is considered to be extremely dangerous. The World Health Organization (WHO) rates it as a risk group 4 pathogen, requiring biosafety level 4 equivalent containment. In the United States, the NIH, National Institute of Allergy and Infectious Diseases, ranks it as a Category A priority pathogen, and the Centers for Disease Control and Prevention lists it as a Category A bioterrorism agent. It is also listed as a biological agent for export control by the Australia Group. The virus can be transmitted by exposure to one species of fruit bats, or it can be transmitted between people via body fluids through unprotected sex and broken skin. The disease can cause bleeding, hemorrhage, fever, and other symptoms similar to Ebola. However, Marburg virus is not the same as Ebola, although similar. According to the WHO, there are no approved vaccines or antiviral treatment for Marburg. But early, professional treatment of symptoms like dehydration considerably increases survival chances. In 2009, expanded clinical trials of an Ebola and Marburg vaccine began in Kampala, Uganda. History. Discovery. Marburg virus was first described in 1967. It was discovered that year during a set of outbreaks of Marburg virus disease in the German cities of Marburg and Frankfurt and the Yugoslav capital Belgrade. Laboratory workers were exposed to tissues of infected grivet monkeys. The African green monkey, Chlorocebus ethiops, at the Bering work, a major industrial plant in Marburg which was then part of Herscht, and later part of CSL Bering. During the outbreaks, 31 people became infected and 7 of them died. Nomenclature. The virus is one of 2 members of the species Marburg virus, which is included in the genus Marburg virus, family Filoviridae, and order Mononagavirales. The name Marburg virus is derived from Marburg, the city in Hesse, Germany, where the virus was first discovered, and the taxonomic suffix virus. Marburg virus was first introduced under this name in 1967. The virus name was changed to Lake Victoria Marburg virus in 2005. Confusingly making the only difference in distinguishing between a Marburg virus organism and its species is a whole italicization, as in Lake Victoria Marburg virus. Still, most scientific articles continued to use the name Marburg virus. Consequently, in 2010, the name Marburg virus was reinstated and the species name changed. Virology. Genome. Like all Mononaga viruses, Marburg virions contain non-infectious, linear non-segmented, single-stranded RNA genomes of negative polarity that possess inverse complementary 3' and 5'. Termini. Do not possess a 5' cap, are not polyadenylated, and are not covalently linked to a protein. Marburg virus genomes are approximately 19 kbp long and contain 7 genes in the order 3, UTR-NPVP350GPVP30VP24L5, UTR. Structure. Like all filoviruses, Marburg virions are filamentous particles that may appear in the shape of a shepherd's crook or in the shape of a U, or a 6, and they may be coiled, toroid, or branched. Marburg virions are generally 80 nanometers in width, but vary somewhat in length. In general, the median particle length of Marburg viruses ranges from 795 to 828 nanometers. In contrast to Ebola virions, whose median particle length was measured to be 974–1086 nanometers. But particles as long as 14,000 nanometers have been detected in tissue culture. Marburg virions consist of 7 structural proteins. At the center is the helical ribonucleocapsid, which consists of the genomic RNA wrapped around a polymer of nucleoproteins. NP. Associated with the ribonucleoprotein is the RNA-dependent RNA polymerase L, with the polymerase cofactor VP35, and a transcription activator VP30. The ribonucleoprotein is embedded in a matrix formed by the major VP40 and minor VP24 matrix proteins. These particles are surrounded by a lipid membrane derived from the host cell membrane. The membrane anchors a glycoprotein GP1, 2, that projects 7 to 10 nanometers spikes away from its surface. While nearly identical to Ebola virions in structure, Marburg virions are antigenically distinct. Entry: Niemann-Pick C1. NPC1. Cholesterol transporter protein appears to be essential for infection with both Ebola and Marburg virus. 2 independent studies reported in the same issue of Nature showed that Ebola virus cell entry and replication requires NPC1. When cells from patients lacking NPC1 were exposed to Ebola virus in the laboratory, the cells survived and appeared immune to the virus, further indicating that Ebola relies on NPC1 to enter cells. This might imply that genetic mutations in the NPC1 gene in humans could make some people resistant to one of the deadliest known viruses affecting humans. The same studies described similar results with Marburg virus, showing that it also needs NPC1 to enter cells. Furthermore, NPC1 was shown to be critical to filovirus entry because it mediates infection by binding directly to the viral envelope glycoprotein, and that the second lysosomal domain of NPC1 mediates this binding. In one of the original studies, a small molecule was shown to inhibit Ebola virus infection by preventing the virus glycoprotein from binding to NPC1. In the other study, mice that were heterozygous for NPC1 were shown to be protected from lethal challenge with mouse-adapted Ebola virus replication. The Marburg virus life cycle begins with virion attachment to specific cell surface receptors, followed by fusion of the virion envelope with cellular membranes and the concomitant release of the virus nucleocapsid into the cytosol. The virus RDRP partially uncoats the nucleocapsid and transcribes the genes into positive stranded mRNAs, which are then translated into structural and non-structural proteins. Marburg virus L binds to a single promoter located at the 3' end of the genome. Transcription either terminates after a gene or continues to the next gene downstream. This means that genes close to the 3' end of the genome are transcribed in the greatest abundance, whereas those toward the 5' end are least likely to be transcribed. The gene order is therefore a simple but effective form of transcriptional regulation. The most abundant protein produced is the nucleoprotein, whose concentration in the cell determines when L switches from gene transcription to genome replication. Replication results in full-length positive stranded antigenomes that are in turn transcribed into negative stranded virus progeny. Genome copies. Newly synthesized structural proteins and genomes self-assemble and accumulate near the inside of the cell membrane. Virions bud off from the cell. Gaining their envelopes from the cellular membrane they bud from. The mature progeny particles then infect other cells to repeat the cycle. Ecology. In 2009. The successful isolation of infectious MAV was reported from caught healthy Egyptian fruit bats. Raucetus aegyptiacus. This isolation, together with the isolation of infectious RAVV, strongly suggests that Old World fruit bats are involved in the natural maintenance of Marburg viruses. Further studies are necessary to establish whether Egyptian raucettes are the actual hosts of MAV and RAVV or whether they get infected via contact with another animal and therefore serve only as intermediate hosts. In 2012, the first experimental infection study of Raucetus aegyptiacus with MAV provided further insight into the possible involvement of these bats in MAV ecology. Experimentally infected bats developed relatively low viremia lasting at least 5 days, but remained healthy and did not develop any notable gross pathology. The virus also replicated to high titers in major organs, liver and spleen, and organs that might possibly be involved in virus transmission: lung, intestine, reproductive organs, salivary gland, kidney, bladder, and mammary gland. The relatively long period of viremia noted in this experiment could possibly also facilitate mechanical transmission by blood-sucking arthropods in addition to infection of susceptible vertebrate hosts by direct contact with infected blood. Evolution. The viral strains fall into 2 clades: Raven virus and Marburg virus. The Marburg strains can be divided into 2, A and B. The A strains were isolated from Uganda, 5 from 1967, Kenya, 1980, and Angola, 2004–2005, while the B strains were from the Democratic Republic of the Congo epidemic, 1999–2000, and a group of Ugandan isolates isolated in 2007–2009. The mean evolutionary rate of the whole genome was 3.3 times 10⁻⁴ substitutions, site per year. Credibility interval 2.0 to 4.8. The Marburg strains had a mean root time of the most recent common ancestor of 177.9 years ago. 95% highest posterior density 87 to 284. Suggesting an origin in the mid-19th century. In contrast, the Raven strain's origin dated back to a mean 33.8 years ago. The early 1980s. The most probable location of the Marburg virus ancestor was Uganda, whereas that of the RAVV ancestor was Kenya. Human disease. MAV is one of 2 Marburg viruses that causes Marburg virus disease, MVD, in humans. In the literature also often referred to as Marburg hemorrhagic fever, MHF. The other one is Raven virus, RAVV. Both viruses fulfill the criteria for being a member of the species Marburg virus because their genomes diverge from the prototype Marburg virus or the Marburg virus variant Musoke MAV, moose, by less than 10% at the nucleotide level. Recorded outbreaks. Prevention. The Public Health Agency of Canada has a candidate RVSV vaccine for Marburg virus. RVSV-MAV. Merck was developing it, but discontinued development. Biological weapon. The Soviet Union had an extensive offensive and defensive biological weapons program that included MAV. At least 3 Soviet research institutes had MAV research programs during the Cold War. The Virology Center of the Scientific Research Institute for Microbiology in Zagorsk. Today Sergeyev Posad. The Scientific Production Association, Vector. Today the State Research Center of Virology and Biotechnology, Vector, in Koltsovo, and the Irkutsk Scientific Research Anti-Plague Institute of Siberia and the Far East in Irkutsk. As most performed research was highly classified, it remains unclear how successful the MAV program was. However, Soviet defector Ken Alibek claimed that a weapon filled with MAV was tested at the Stepnogorsk Scientific Experimental and Production Base in Stepnogorsk, Kazakh Soviet Socialist Republic, today Kazakhstan, suggesting that the development of a MAV biological weapon had reached advanced stages. Independent confirmation for this claim is lacking. At least one laboratory accident with MAV, resulting in the death of Koltsovo researcher Nikolai Ustinov, occurred during the Cold War in the Soviet Union and was first described in detail by Alibek. MAV is a select agent under U.S. law. References. Further reading. External links. International Committee on Taxonomy of Viruses. ICTV. FILOVIR. Scientific Resources for Research on Filoviruses.